welcome to episode 10 of the CASK podcast, and my name is Laura Hattersley, and today I'm going to talk about the very exciting news that has come from a lab in Japan about a potential drug treatment for our children. So Professor Tabuchi has been studying CASK for many years. He's probably one of the world's most experts on CASK disorders, and recently finished his kind of set of experiments that he'd been doing. And although his work hasn't been officially published, it is in preprint because it is awaiting reviewers for one of the journals that he has submitted it to. So it can be read, all his results, and we also have a summary of the results. But I'm just going to talk a little bit about what he's found, some of the background to it, and what this could mean for our community. So, as we know, most of our children have microcephaly, which means that their brain is smaller. And, we've always known this. This is one of the main ways that you first get down that diagnosis process to find out if they actually have a CASK gene disorder. So it's a very common symptom. And obviously, the first question that should come to mind is, why are their brains small? Which part of their brain is small? And what is causing the brain to be small? And this is something that scientists have been looking into for some time. And obviously MRIs can show which parts of the brain are small. So we know that a lot of our children have MIC-PCH. And that actually comes from the MI is for the microcephaly part of the brain, and the PCH is a description of what the brain looks like. So pontocerebellar hyperplasia, ponto for the P, cerebella for the C, and hyperplasia for the H. And what this means is small pons and small cerebellum. So that we know that this is the phenotype, the anatomical phenotype that our children have. And scientists like Dr. Tabuchi have obviously been trying to work out, well, what is causing this? Like, we know it's the CASK gene, but what is the lack of CASK protein doing to create this kind of deficit in brain size? And Dr. Tabuchi's lab has discovered that the pathway and the exact mechanisms of the cause. So I have to say, I won't give Dr. Tabuchi all the credit, because there have been some other incredible scientists who are still working on CASK, and it's like the collaborative effort of these scientists bringing out work in their separate labs, but kind of creating this whole picture of what we now know about CASK. So Professor Mukherjee over in the US has published quite a few papers which show that the cells in the brains are dying. It's not that they're not being made, it's that they're dying. And I think this was kind of big news, really, within the scientific community and for the parents if they heard this news, because that then took us down that path of, well, what is killing the cells? So that's a very different treatment options than if the cells just weren't being made in the first place. So following that, obviously the researchers have now been looking, and I know Professor Mukherjee is also looking at drugs that can target this. So scientists have been looking at, well, what is killing these cells? So it's the cerebellar cells that they've been looking at mostly. So cerebellar granule cells are dying, and the cerebellum is a large part of the brain, and if the cells are dying in it, obviously cerebellum is smaller, and that's one of the major reasons why our children have microcephaly. So Dr. Tabuchi has discovered, and I will keep the science succinct, hopefully, he has worked out that if there is no CASK, so like in our children, or no CASK in particular cells, we know that in females with MIC-PCH, some of their cells do have the CASK protein and some don't. But in the if there is no CASK in particular tissues or cells, then that leads to quite a destructive process that then leads to something called programmed cell death, which is the correct term for that is apoptosis. And it's all kind of masterminded by a molecule or compound called JNK. So when the area in the brain is CASK deficient, JNK gets activated, and it activates this destructive pathway, part of which is you get something called reactive oxygen species, and that then kills the cells. So obviously knowing this leads to the idea of, okay, how can we then treat it? So Dr. Debuchy actually took an inhibitor. There were already known inhibitors of JNK, obviously called JNK inhibitors, and he looked at quite a selection of these and tested, I can't remember now, maybe 5 or 6 different inhibitors in cell cultures, and they all they all had an impact, a positive impact, and seemed to stop the cell death. And then he chose the one that seemed to have like the most potential, and he's tested those in mice, and again it has had an impact. So it's seems to have inhibited the JNK pathway, so it has stopped that cell death. And you can see in the mice that the brain cells are not dying, or at least not dying at the same rate. And kind of most excitingly, that translates into improved symptoms within these mice. So obviously it's quite hard to measure everything in a mouse that might be relevant to a human, but there are certain kind of standardized tests that they do with mice. So one is like limb clasping. So when mice often have developmental disorders, they kind of clasp differently with their limbs. So he measured that, and also their gait. So just kind of very similar to our children, really. Mice with MIC-PCH have a irregular gait, and so that's obviously like their walking pattern, and you can easily measure that. So he used those things as measurements, and found, like really improved symptoms. So all that data gives a really nice idea that actually this drug could potentially be, lead to similar symptom improvements in humans. Now, I just want to say, if anybody has had a good look at the paper, the mouse results are in females, but that does not mean that this wouldn't be applicable or helpful for males. He only does it in females because sadly males given MIC-PCH, male mice, they don't survive after birth. I think they die on day 0, day 1. So that's the only reason, because it's just not, you just can't work on the dead mice. So this is a potential therapy that could help both sexes. We don't obviously know how much of the human symptoms are the result of the small cerebellum and the cell death, but obviously we do know a lot about what the cerebellum does. And the cerebellum is involved in coordination, balance. So probably, the movement issues we see with our children are very, very likely down to the fact that they have a smaller cerebellum. But it's also involved in emotion and lots of other things. So, it could be by being able to treat this particular aspect, it could actually have quite wide-ranging impacts. So it's really exciting. And CASK research and the CASK coalition in general. So, I should add that Dr. Debuchy is actually the advisor for our partners in Australia, ACNRF. And so this is how we found out about it, because we all chat to each other and let each other know what's going on. And obviously, if I thought I might be able to help originally with some funding avenues that are here in the UK. And we're still investigating that. So I kind of offered to help, and we're now looking to see how we can take these exciting findings and hopefully move it down that pathway towards an actual therapeutic. And I'm no expert in this, so I've reached out to various organisations, various people. I'm very busy at the moment with a lot of meetings. So if anybody wants to support us, that would be great. Even any volunteers, as I always say, it just takes some of the workload off me, so I can concentrate on, for example, having meetings with pharmaceutical companies and experts in this field. So please do get in touch if you would like to volunteer. And we are right at the end, so I think I'm going to leave it at that. But thank you for listening, and hope everybody's well.